It's a pleasure to be here and share with some of the results that we've been working on, and I'll just introduce the topic a little bit, where we've been focusing and why we've been focusing, where we're putting our efforts, and then turn it over to Zoe and Jamie to share with you the details of some of the studies and the results and where we're at, and then when we're having the open discussion and panel, really looking forward to hearing what you think about the results and how we plan and look forward to go towards the next stage. So, many of you might be familiar with a project that we did that was funded by the National Pork Board, and several in the room here were really drivers of helping establish the availability of that funding to do a survey of the industry to try to better understand the distribution and causes of pelvic organ prolapse. And during that course of that survey, we collected all kinds of information on herd factors, facility factors, management, and then animal-based measures, and really to try to get our head around what are the potential items or potential biological underlying causes that could be contributing to pelvic organ prolapse, so that we could do a more hypothesis-driven research approach to better understand the problem. In that project and in that survey, we worked with 104 different sow farms across the United States that were in 15 different states in the U.S. There were 85 farms from larger systems, production systems, 19 from independent farms, and average sow inventory from 600 to 10,000. And we had a lot of different things that we looked into, and all that information is on our National Pork Board final report that's on the Pig Livability website. But one of the things that we verified was that 21% of all the sows that are dying in U.S. farms was due to pelvic organ prolapse. And that number on some farms now in the last 2 years has continued to increase for many of the producers and farmers that we work with and interact with. One of the things, though, that I think was one of the biggest takeaways of that survey is that when we visited 62 different sow farms, we had created and used a pelvic perineal scoring system to evaluate the perineal area and assess whether we could use a scoring system to predict risk for prolapse in those sows. And so it was a three-point scoring system. 1 was a little to no risk of prolapse, 2 was a moderate risk of prolapse, and 3 was a high risk of prolapse. And in that initial study, when we collected data that we got back from the farms the following 2 to 3 weeks, when we looked at the identities of those sows that died due to pelvic organ prolapse, we were able to confirm that perineal score 3 had a substantially higher rate of dying due to POP than the sows that were a 1 or 2. And I think that really was fundamental for us, the way we think about it, because it gave us a strategy to collect biological samples from sows that had a higher probability of prolapsing compared to sows that did not. And even though it's a dramatic and a significant issue to the industry, in some farms that struggle with pelvic organ prolapse, sometimes it's only 3 to 5, maybe 6 sows a week are dying, which is really hard to design experiments around only sows that actually experience POP. So the perineal scoring system has been very, very important. The average perineal system or average perineal score for a sow farm is correlated to the percentage of sows that die on that farm due to pelvic organ prolapse. And it was, again, a demonstration that something biologically is different in those sows that is not present in sows that have a low risk or a low perineal score. So that's really the basis of Zoe and Jamie's experiments that they're going to share with you, is how can we try to exploit that scoring system to better understand the biology. And that's how we've designed some of the experiments to understand it, and then also use it as a gauge to evaluate mitigation strategies. So, with respect to the current grant that we're in, the work that Zoe and Jamie are going to share with you is part of Research Objective 2, identification of putative mortality causes in U.S. farms, and coupled with the development and implementation of strategies to maximize survivability. So I'll turn it over to Zoe to share some of her work in 2.2a.
Sorry. All right, so, do the slides come back on? Okay. So like Jason mentioned, a lot of our work is focusing on the current grant, project number 2. And so a lot of the work that I've been working on is specifically relevant to the physiological, endocrinological, and microbial interactions and how that influences POP. So dysbiosis is an imbalance or decrease in the microbial diversity and has been shown in humans suffering from pelvic inflammatory disease or bacterial vaginosis, as well as in cattle suffering from endometriosis or other uterine diseases. However, at the beginning of this grant, there was really little to no knowledge on swine vaginal microbial community or linked to any sort of reproductive health in general, which is why we kind of decided to work on that. And so in our first study, we wanted to evaluate the vaginal microbiota and serum metabolites and differences in the late gestation commercial sows at varying risk for pelvic organ prolapse. And we went on 2 different commercial sow farms with different producers, different states, and collected samples from about 213 animals, including perineal score. And so you can see that, like Jason mentioned, the perineal score is an effective measurement in determining prolapse risk, and our perineal score 3 animals had a significantly higher chance of prolapsing than our perineal score 1. And like I mentioned, we collected vaginal swabs and blood samples from perineal score 3 animals parity matched to a perineal score 1. So with those vaginal swabs, we evaluated the microbial communities and saw differences on a whole community level between perineal score 1 and perineal score 3 sows. And when evaluating just the top 100 most abundant microbes, we saw that there were 24 that were significantly different between perineal scores. So 12 of those were increased in our perineal score 1 sows, and I've highlighted a few that we think are of interest on the slide here, and as well as 12 were increased in our perineal score 3 sows. So Streptococcus dysgalactica is a known pathogen and has been linked to pelvic inflammatory disease and bacterial vaginosis in humans, as well as equine reproductive infections. Treponema is also linked to inflammation in other species and reproductive health, as well as swine dysentery, which is an inflammatory bowel disease. And then we have Prevotella, which again is a known pathogen and has been shown to have negative effects in other species. So in addition to the vaginal microbiota work, we did GCMS on our serum samples and identified 93 molecular features, and out of those 93, 16 of them were significantly different between our perineal scores. And so interestingly, all of those were increased in our high-risk animals, so our PS3 animals. And a couple of interests include D-fructose, which is a derivative of glucose and has been shown to change in circulating levels in women suffering from bacterial vaginosis or other reproductive dysfunctions, as well as 2-aminobutonic acid, which is a derivative of butyrate and has been shown to be correlated with inflammation. And butyrate is a microbial-produced short-chain fatty acid. So then that kind of ties us into our second study. Again, we wanted to validate those results from the first study and replicate it on a larger scale. And so the objective of this study was to determine sow vaginal microbiota differences and changes in late gestation and associated with POP. And this one differs from the first study as it was on 2 larger commercial sow farms, same producer, same state, within a mile of each other, same feed mill, GDU, as similar as we could get them, except for them being 2 different farms. And interestingly, we were able to observe similar changes between the 2 studies. So a little bit into the methods, animals were scored once a week throughout gestation week 12 through 15. And then you can see here that at gestation week 12, a lot of your animals are going to be scored that PS1 or low risk. And then as parturition looms, you'll see that increase in the red bar is indicating the perineal score 3 animals. So then at gestation week 15, we collected vaginal swabs. And then you can see again, validating our perineal score system, that the majority of animals scored a perineal score 3 are going to prolapse compared to only 1% of the perineal score 1 animals. So with this study, just over 50 OTUs were able to be identified as significantly different when evaluating the top 100 most abundant OTUs between perineal score. So 18 of those were more abundant in our PS1 animals, including Porphyromonas and Enterococcus, which were similar in our previous study, being increased in our low-risk animals. And 33 of those were increased in our perineal score 3 sows, including Clostridium, which has been shown to be increased in, again, human reproductive dysfunction, as well as several other livestock species. Duncanella, which is in the Prevotella family, and Prevotella has been shown to degrade mucin and increase gut permeability, and in the reproductive tract, mucin acts as a barrier between pathogens, so could have a role playing in there, as well as Streptococcus dysgalactia and Treponema, which we saw to be increased in our PS3 animals in the previous study, as well as Staphylococcus, which is an emerging pathogen in poultry and has been shown to have an effect on collagen, which plays a large role in the reproductive tract and ligaments. So in addition to just evaluating the differences between our high and low risk animals, we wanted to then take a closer look at our high risk animals and see if there were any differences that could be observed between those that did or did not subsequently experience prolapse. And as you can see here, only 2 were significantly different, and both of those were increased in our high risk animals that did not subsequently experience prolapse. And Veillonella could be a candidate of interest because it was significantly increased in our PS1 animals compared to PS3 in our initial study, and there tended to be a difference in this larger study between perineal scores. In addition to microbial differences associated with POP, we also wanted to look at other physiological and endocrinological differences could be observed. So the objective of this third study was to identify any other putative biological factors that could be associated with POP risk. So similar to the previous study, we used the same set of animals scored once a week throughout gestation week 12 and 15. And at gestation week 15, we collected blood samples that we sent for CBC analysis, evaluated for biomarkers, and also performed a steroid hormone analysis. And again, just a reminder, the high risk are PS3s, low risk PS1s. And so in our CBC analysis, we saw decreases in immune cell populations within our perineal score 3 sows, and we saw differences in mean platelet volume, monocytes, and lymphocytes. And MPV has been associated with inflammation, and lymphocytes and monocytes are also a known marker for inflammation. And interestingly, they're all decreased in PS3 cells, which could in part be explained by a reallocation of those immune cells to target tissues, which is why we're not seeing them in circulation. They could be going to that source of inflammation, and monocytes specifically are known to be pro-inflammatory and recruit other immune cells to that target tissue. In addition to our CBC work, we looked at several different biomarkers of inflammation and found that LPS binding protein was significantly increased in our perineal score 3 sows, and LBP is a known inflammatory marker and is known to be associated with bacterial endotoxins, so kind of playing a role in what we've seen in the differences in the microbial communities. In addition to the biomarkers of inflammation, steroid hormones have also been shown to fluctuate in association with inflammation or infection. And in our animals, we saw differences in 4 of the steroid hormones. And specifically, estrogen is of interest because it has been shown to be increased in women suffering from POP or endometriosis, as well as has been shown to correlate with inflammation and have a pro-inflammatory response. So Over the last few years, what have we learned to be associated with this high risk for prolapse? So we know that there was some sort of biological changes happening with these sows that makes them different from the low risk. We found that there were changes in the vaginal microbiota as well as serum metabolites, and specifically looking at the vaginal microbiota across these 2 studies, we were able to find microbes of interest that either had the same trending, being increased or decreased in our perineal score 3 animals, as well as a handful of microbial species that were abundant in both species, which can help us better define the sow vaginal microbiota healthy community and figure out what exactly a dysbiosis looks like within that animal. Additionally, we found decreases in immune cell populations within our perineal score 3 sows, increases in circulating biomarkers, as well as increased steroid hormones in our high-risk sows. And additionally, some future work that we already have planned in relationship to the microbiome is we would like to evaluate the fecal microbiota of those sows at differing risk for POP. So those animals in study 2, in addition to the vaginal swabs, we did collect fecal swabs and are working on analyzing that, as well as doing some metagenome shotgun sequencing on the vaginal microbiome, and that will give us more functional data, so not just necessarily what microbes are there, but what those microbes are doing and what they're producing. And another goal of this grant was to look at mitigation strategies and testing the efficacy of those, and so Jamie is going to talk a little bit more on that.
So following Zoe's work on the vaginal microbiome, we decided to pursue research into the area of antibiotic usage as a mitigation strategy to reduce prolapse incidence. And from the initial survey, they discovered that prolapse incidence was decreased in the period after or during an antibiotic pulse compared to the weeks following the antibiotic pulse, so just comparing the weeks surrounding an antibiotic pulse. And this is all just initial survey data. So then for our study, we partnered with Dr. Daniel Boykin and Cactus Family Farms to conduct a study to evaluate the effects of BMD supplementation on prolapse incidence in late gestation sows. And BMD is a narrow spectrum antibiotic, and it's used in sows to control Clostridial enteritis, which is caused by Clostridium perfringens. So for our study, we treated sows with BMD for 2 weeks pre-farrow, and at gestation week 14, we would allocate sows into BMD treated or control groups, and then at one time point during gestation week 15, they were assigned a perineal score. And then this study was conducted at 2 sow farms. So at farm A, BMD was administered through the water, and then at farm B, BMD was administered as a top dress. And then both of these farms were utilizing BMD in their lactation diets, but we were administering treatments for 2 weeks pre-farrow, so while they were still in gestation. So I analyzed the data from each farm separately. So at farm A, BMD was in the water, and at farm B, it was administered as a top dress, so I just wanted to remind you of that. But BMD treatment didn't affect the percentage of sows scored a perineal score 1, 2, or 3 at either farm. And then the overall outcome, the main outcome of the study was that BMD treatment did not affect prolapse incidence at either farm. So at farm A, 2.7% of the control sows and 2.8% of the BMD treated sows prolapsed, and then at farm B, 2.8% of the control sows and 3.2% of the BMD treated sows experienced a prolapse. But then when looking at the litter characteristics in sow performance, we did observe a tendency for an increase in the number of live-born piglets at farm B in the BMD treated group, but then this can likely be explained by this decrease we see in the number of stillborn piglets in the BMD treated sows, and this is observed on both farms. And then when looking at the number of stillborns by treatment and perineal score, you can see that this decrease in stillborns is in the perineal score 3 BMD treated sows compared to the perineal score 3 control sows, and this is observed on both farms. So overall conclusions from this study, BMD treatment did not affect perineal score or prolapse incidence, but we did observe a decrease in the number of stillborn piglets in the BMD treated sows compared to control. So thank you for listening to our talk this morning, and I'm sure we don't have time for questions, but we'll be here for the open session to answer questions.